synthesis is the process of linking amino acids together to form a polypeptide. Transcription is the synthesis of RNA under the direction of DNA, and it produces mRNA, which is messenger RNA. That takes place in the nucleus. Translation is the actual synthesis of the polypeptide, which occurs under the direction of mRNA. It's on ribosomes.
The initiation of transcription is regulated by the transcription factors that attach to the promoter region, which is right over here, as you can see. Then the RNA polymerase binds to the promoter. The DNA strands unwind, and the polymerase initiates RNA synthesis at the start point of the template strand. And you have the Tata box right here. Yes, it's called the Tata box. RNA polymerase. It's this big yellow thing. Transcription factors. So, it's pretty good.
RNA polymerase moves along the DNA and it untwists the double helix, showing the DNA bases that pair with the RNA nucleotides that come in according to their base pairing rules. And then RNA polymerase 2 deciphers that sense DNA strand and adds the nucleotides from the 5' to 3' direction.
Termination of transcription occurs when RNA polymerase the second reaches a DNA stop region or termination sequence AAU AAA in the DNA. Then RNA polymerase jumps off the DNA and the mRNA peels completely free from the sense DNA. And as you can see in the picture, that's what's happening.
mRNA processing must occur before the mRNA leaves the nucleus. The 5' is a modified guanine cap and adds to the 5' end of the mRNA. The 3' poly A tail is added to the 3' end. They also help the ribosomes attach to the 5' end of the mRNA. And then that's what's going on the top, is that they go from different stages. What else is going on is called mRNA splicing, which is the introns from the pre-mRNA. The pre-mRNA consists of extrons and introns. And the exons are the coding regions that code for amino acids, and introns are the non-coding regions that don't code for the amino acids, so they're the opposite. And they must be removed from the pre-mRNA before translation or else it won't work. The mRNA is edited by spliceosomes, and what those consist of they're called SNRPs. And then what combines with the SNRPs are other types of proteins. The SNRPs attach to each of the ends of the intron, while the other SNRPs pull the ends of the intron. Together, when they connect, they form a spliceosome, and it removes the introns, so the extrons can take its place.
Hi, I'm Hunter, and we use the mRNA coding dictionary to determine which mRNA codon codes for which particular amino acid. Example, the mRNA codon CGA codes for arginine. The codon AUG not only codes for the amino acid methionine, but also indicates the start of translation.
So computer translation is the actual synthesis of the polypeptide, which occurs under the direction of the mRNA. It's on the ribosomes. The ribosomes have 3 sites, the A, P, and E sites, which bring in the amino acid chain and link amino acids to each site and build a protein off of codons. And there's 2 subunits, the top part and bottom part of the ribosome.
A tRNA molecule. To form a 3-dimensional structure, our tRNA molecules are made along genes in the nucleus by RNA polymerase. It includes a loop containing the anticodon and an attachment site at 3' end for an amino acid.
The amino acid tRNA synthetase is a special enzyme, in cytosol that connects to the proper tRNA to the correct amino acid. So these two have to connect back into here. So that's what this one shows. And so once it finds the correct amino acid, they bind together. And there's 20 different forms of this kind of enzymes. It's 20 for each of the different amino acids.
The initiation of translation begins with the 5' end of mRNA attaches to the small ribosomal subunit at the start codon. Next, the initiator tRNA carrying methionine amino acid with the anticodon UAC base pairs the start codon in the P site. Finally, the large ribosomal subunit, right here, attaches. The arrival of the large ribosomal subunit completes the initiation complex.
Elongation of translation from the cycles amino acids are added to the preceding ones to create a protein, and those are created by codons. The A site is empty, so the tRNA molecule carrying out the second amino acid lands and fills it up, and that's comes in and they attach. The tRNA anticodon must be complementary to the mRNA codon in the A site to make sure the correct amino acid is being added. And then peptide bond formation occurs as a bond between the peptide and the P site and joins the new amino acid in the A site.
During translocation, the ribosome moves tRNA with the attached polypeptide from the A site to the P site. The tRNA that had been in the P site is moved to the E site and then leaves the ribosome. Translation ensures that the mRNA is read 5' A 3' codon by codon. It takes 1/110 of a second for this process.
Termination of translation occurs when the ribosome reaches the stop codon, for example, such as UAG, UAA, or UGA, so that happens on the mRNA. And then at the A side of the ribosome, it accepts a protein, and which is called the release factor. And so what the release factor does is it hydrolyzes the bond, and then the P site of the amino acid is the polypeptide chain.
Amino acid consists of an amino group, a carboxyl group, a central carbon, and a R group. Every amino acid is exactly the same except for the R group.
The rRNA is large ribosomal subunit acts as a ribozyme to join amino acids to form a peptide bond. A bond forms between the carboxyl end of the amino acid and the amino group of the next amino acid. Water is removed as the bond is formed.
The polyribosomes consist of many ribosomes translating a single mRNA molecule simultaneously. This allows proteins to be formed quickly.
There are 4 levels of the protein structure. The first is the primary structure, which is a sequence of amino acids in the polypeptide chain, and those are determined by the DNA. The secondary structure is just a bunch of loops and coils that form by the hydrogen bonds by they're non-adjacent, the carboxyl and amino groups. The tertiary structure is the 3-dimensional form that's held together by the interaction between R groups and water because of the electric charges. And then this is the quaternary structure, which is the last one.
After translation of the polypeptide chains undergo modifications, this takes place in the Golgi apparatus, which is this thing, that Wilcher. Sugars, lipids, and phosphates are added onto some amino acids. Proteins may also be modified in ways that affect their 3-dimensional shape. And yeah, that's it. This is the end of our project. We all worked hard on this project, so sorry, Ms. Silva, if it isn't as good as you expected it. But I think we at least deserve a B. And don't get mad at Will, because even though he wasn't here,
He did some good stuff. Good job, Will. Love you. All right. Peace out.